Hello and welcome. My name is Cecilia Canarozzo and I'm a PhD student in Aerocastren's lab at the University of Helsinki. Today I will introduce you to our work recently published, where we have investigated the presence of cholesterol recognition motifs in the transmembrane domain of the tyrosine kinase receptor family, with a particular focus on TRKB. Let's start with a brief introduction about the role of cholesterol in the brain. The cholesterol present in the brain accounts for 23% of the total cholesterol in our body. Of this 23%, the majority of the cholesterol is present in the myelin sheaths of oligodendrocytes. Cholesterol does not pass the blood-brain barrier, therefore it needs to be locally synthesized. During early stages, neurons are able to synthesize cholesterol, and this synthesis peaks at the second postnatal week, followed by a decrease in production by the neurons and taking over of the synthesis by the astrocytes. So the astrocytes become the main source, and cholesterol gets transported to the neurons through an ApoE-mediated mechanism. Cholesterol has several functions. It promotes neuronal maturation, proper synaptic transmissions. It's responsible for several physical properties of membranes, such as curvature and fluidity. It is a core constituent of lipid rafts, which act as signaling platforms for several pathways. And it interacts with transmembrane domains to regulate protein functions. So how does cholesterol interact? From literature, we know that cholesterol can interact with other molecules thanks to several consensus motifs. And here we're going to focus on two types: the cholesterol recognition amino acid consensus CRAC and its inverted version CARC. The linear sequence of CRAC from the N-terminal consists of an apolar residue, leucine or valine, 1 to 5 amino acid of any kind, a central aromatic residue, a tyrosine or a phenylalanine, which seem to be also the most important for the interaction with cholesterol, 1 to 5 amino acid of any kind, and a basic residue, which can be an arginine or a lysine. CARC motifs have the same sequence, but it's inverted. And they can also have a tryptophan as a central aromatic residue. It's also been shown that CARC motifs have a stronger affinity for cholesterol. So the transmembrane domains of protein can present CARC and CRACs, or both together, with the CARC present in the outer leaflet and the CRAC present in the inner leaflet. Through these motifs, cholesterol can interact with the transmembrane domains of proteins, and therefore can promote receptor dimerization by bringing closer together the monomers of the receptor. One family of receptor that needs the dimerization to work is the tyrosine kinase receptor family. When the ligand binds the binding site on the extracellular side of the receptor, this binding stabilizes the homodimerization, which induces the trans-autophosphorylation. So each monomer phosphorylates the other in specific tyrosine residues in the intracellular domain. So our question was, do these receptors interact with cholesterol? Do they have these CARC motifs? To answer this question, we searched for the RTK family in UniProt. We looked at different species. We chose the curated info and extracted the transmembrane FASTA sequence for each protein. And finally, we looked for CRAC and CARC motifs. We found that CRAC motifs are shown both in vertebrates and in invertebrates, whereas we found that CARC motifs are present only in vertebrates. Here is a list of members of RTKs that have either the CARC or CRAC motifs in the transmembrane region. And in particular, we found a CARC motif in the transmembrane region of TRKB. Only a CARC, no CRACs. And we focused our further investigation on TRKB since our laboratory is focused mainly on this tyrosine kinase receptor, which is the receptor for BDNF, the brain-derived neurotrophic factor. So what is the relationship between TRKB and cholesterol? Well, upon BDNF binding, the activated TRKB translocates to lipid rafts, which are microdomains in the membrane that are enriched in cholesterol. From literature, we also know that BDNF promotes the production of cholesterol, and at the same time, cholesterol regulates TRKB signaling. So after defining the presence of a CARC motif in the transmembrane region of TRKB, we investigated further, starting with the percentage of identity between different species. Here you can see the transmembrane sequence of TRKB of different species with the CARC motif underlined in highlighted in red. And you can see how conserved this is. In fact, we found that we found over 90% of identity in the amino acid sequence from different species of both the transmembrane region of TRKB and full-length TRKB, as can be seen in these trees. We also found a significant correlation between the percentage of identity of the full-length TRKB and the transmembrane region of TRKB. So this data suggests that this motif and TRKB have evolved together. We then asked our collaborators to investigate the interaction of this motif with cholesterol using molecular dynamic simulation. These are snapshots of the transmembrane region created for the simulation. We can see the interaction between the transmembrane region of TRKB and cholesterol. In particular, we can see that cholesterol interacts with the hydroxyl group of the central tyrosine 433 in the CARC motif of the transmembrane region of TRKB. What the simulation showed is that the average occupancy of cholesterol molecule is much, much higher for the C-alpha atom of the tyrosine Y433, but not for the C-alpha atom of the arginine 427. And this confirms that the aromatic tyrosine is central for the interaction of TRKB with cholesterol. We then moved in vitro and transfected HEX cells to overexpress a GFP-tagged full-length TRKB. After lysis, we took the samples and exposed the immobilized TRKB to different concentration of cholesterol and different concentration of bioethylated BDNF to run a binding assay. So what we saw is that the binding of the bioethylated BDNF to TRKB varies depending on the concentration of cholesterol added to the samples. Lower amounts of cholesterol facilitate the binding of TRKB, whereas higher amounts of cholesterol, higher concentration, compromise it. This is the curve for the bioethylated BDNF at 1 nanogram milliliter with a different concentration of cholesterol. And we can see even better this kind of bell-shaped response. So the cholesterol here at 20 micromolar facilitate the binding of the bioethylated BDNF, whereas the 50 and 100 micromolar of cholesterol compromise it. We then did a scattering analysis, which showed that the effect of cholesterol are mostly due to differences in the available binding sites for the bioethylated BDNF. So our data suggests that depending on the cholesterol concentration, the orientation of the TRKB dimer changes, and this modulates the exposure of the binding sites for BDNF. Importantly, this is what we saw previously for BDNF-induced TRKB activation, where low levels of cholesterol facilitate the activation of TRKB induced by BDNF, whereas higher concentrations whereas at higher concentration, this increase is lost and compromised. So altogether, the data suggests that this motif might act as a sensor for cholesterol, where TRKB triggers synaptic maturation or neurite growth only if the cholesterol level are optimal. If you want to know more about this story and how antidepressants bind to TRKB, you can check out this paper or the related video on this channel. With this, I would like to thank our lab members and our collaborators for their contributions to our work, as well as our funding agencies for supporting us. And thank you for watching.